Hello. Today I'd like to tell you about my lab, the Pardue Lab. We are developing life-changing treatments for individuals who have vision loss. So my lab is actually located at the Atlanta VA, and I'd like to show you some of the rooms. So one of the first labs that I'd like to show you is a lab that we have that we use for single-cell electrophysiology. So in this room, we have an ePhys rig that is set up that we're using to measure single cell recordings of different retinal cells. You can see that there. This is another one of our rooms. I call this the histology lab. Here we have a space for making different types of solutions. And then on the histology aspect, we have a cryostat and a fluorescent microscope, dissecting microscopes. And then we also have in the back here an ultramicrotome. So this equipment is all used to section eyes and sometimes brains that we use to examine different types of rodent models that are undergoing retinal disease, such as diabetic retinopathy or photoreceptor degenerations. This is another one of our rooms. This is actually a dark room. So I'm walking out of a dark room door into this small room that we use to do electroretinograms. So you can see that we have a Faraday cage, and inside is a system called an electroretinogram. So the platform is actually a heating pad, and it has different holes or plugs where we can plug in electrodes. And so an animal would sit on the heating pad, a mouse or a rat, and then we would put the electrodes on the surface of the eye, on the cornea, and measure a full field potential to a flash of light. This is very useful for our projects when we want to measure retinal function, such in diabetic retinopathy or our photoreceptor degeneration models. We can use it to detect that a disease is there for screening or detection purposes. But then we can also use it to monitor if our treatments and interventions are actually working. And then here, as I look back in the room, you can see that dark room door. This is another room we have, which I call the surgery room. Here you can see a surgical microscope where we might use to do eye injections or look into the inside of the eye. Here we have one of our imaging systems. This is a scanning laser ophthalmoscope and an optical coherence tomography system. So this is used in the clinic, but here we've modified it to be able to use with rodents. So the rodent sits up there on the black and white platform. Then there's a laser that's shined into the eye. And when we have the system turned on, we'll get an image like you can see here of the inside of the eye and the retinal vasculature, which we can use to evaluate how the retinal vessels are functioning or their size and their diameter in diseases such as diabetic retinopathy. So that was the tour of the Pardue Lab. I hope I was able to show you some of the techniques and rooms that we use here in my lab. As I said, our lab is focused on developing treatments, life-changing treatments for individuals who have vision loss. And we do this with a number of different techniques and a number of different approaches. So I hope we have an opportunity to talk further during the interview process. Thank you.